So I wanted to go back to this information because I went into the mitochondrial DNA and now I'm going into what is called junk DNA. What are all these alarms going off? Anyways, it's throwing me off from my studying. Let's read it and then I'm going to get into my thoughts about it. Junk DNA is a dated term for non-coding DNA regions, initially thought useless, but now known that is definitely not a real alarm. What the fuck? Junk DNA is a dated term for non-coding DNA regions initially thought useless, but now known to contain vital regulatory elements controlling gene activity, shaping DNA structures, and influencing health. Its most regulatory sequences, transposons, and pseudogenes makes up 98% of the human genome, challenging the junk label. Hold on. So that was a term from the 1970s. I don't know what they're calling it now because I touched on that before in a video. Current scientific view, the term is largely considered misleading because researchers constantly uncover crucial functions with most non-coding DNA now seen as vital for complex life. So now they're calling it, instead of junk DNA, they're calling it non-coding DNA. What I would say is it's sleeping DNA. It's DNA that's not it's not turned on, right? And we learned that mitochondrial DNA can turn the coding on, right? Let's go down. Where is it? Let's see here. I lost my thing. Where is it? Let's see. That's not what I was looking for. No longer useless, the important roles of junk DNA. The myth that the non-coded DNA sequence have no biological significance has been busted, explains CU research instructor Lane Koningsberg, PhD. Hold on, I gotta make it bigger. I ain't got my glasses on. Let's see if I can see that. When geneticists started mapping the human genome, they were specifically interested in learning about genes and what they do. Everything else they deemed as junk DNA. But as the saying goes, one person's trash is the other person's treasure. So-called junk DNA makes up a vast majority of the genome, about 98%, and consists of non-coded or coding DNA, which scientists now see as vital in studying human health and diseases. Remember when I told you that the mitochondrial Eve, her lineage can be tracked back 400,000 years ago. And they said not because there weren't other women, there weren't other Eves. It's just that her Eve, her DNA stood the test of time. Well, so far it stood the test of time because all of humanity right now has that Eve Eve DNA in them. Sorry, that mitochondrial DNA. Let me go back. There was something I was going to show you. Just a minute. Where is it? Here we go. What is the role of junk DNA in evolution? Their finding published recently in the Journal of eLife indicates that this genetic junk performs the vital functions of ensuring that chromosomes bundle correctly inside the cell's nucleus, which is necessary for cell survival. And this function appears to be conserved across many species. And that's what I wanted to get to. I was going to get into dark matter too, because there is a connection between this so-called turned off DNA and what people call dark matter, or the God matter. It says, why do humans have so much junk DNA? Many of these sequences are the descendants of ancient virus infections and thus non-functional in terms of human genome function. Many sequences can be deleted as shown by comparing genomes. What's the percentage of humans' DNA in junk? About 98% of our DNA is non-coding, often called junk DNA, but this term is misleading. While that less than 2% codes for proteins, the vast majority plays crucial regulatory roles in turning genes on and off, controlling cell structures, and influencing diseases, making it functionally important and not useless. Let's go to gene coding. So remember, let's tie these things together. 98% of your DNA is coded off or turned off. Okay, 2% is turned on to code you as a person. The function of genome coding is to act as a fundamental blueprint for life, storing instructions to build, maintain, and operate an organism by directing the synthesis of proteins and functional RNA molecules, while also encoding crucial regulatory information and even contributing to the physical structure and organization of chromosomes themselves. Cellular machinery, translation, ribosomes read this mRNA in 3 base codons, each specifying a particular amino acid linking them to form proteins. So I'm going to go back to the fact that the mitochondrial DNA is the oldest DNA that has been able to be traced back to a mitochondrial Eve. It's not a mitochondrial Adam, it's a mitochondrial Eve. There is no Adam linked back to that long ago. It's none. So, food for thought.